Good morning everyone. Today we discuss PU questions regarding neural control and coordination. So in this chapter, reflex action, ear, eye are not included. So due to corona effect, they reduced 30% of the syllabus. So in this chapter, these three topics not included. Reflex action, eye and ear. Briefly describe the structure of the following: brain, eye, ear. First question, page number 329. So no need to read eye and ear, only brain. Briefly describe the structure of the following. So you have to describe the structure of brain. We already discussed. Structure of the brain. So structure of brain. Brain is the central information command center. Brain has 100 billion neurons. Brain is the first part of central nervous system. Brain and spinal cord together known as central nervous system. 3 meninges present around the brain and spinal cord. Outer meninge. Outer meninge is dura mater. Middle thin meninge is arachnoid. Inner meninge is pia mater. Central nervous system is protected by 3 meninges only in mammals. In amphibia, reptile saves 2 meninges protect the central nervous system, outer dura mater, inner pia mater. In fishes, only 1 meninge, meninx primitiva. So, presence of arachnoid meninge is the unique character of mammal. The space present between arachnoid and pia mater is known as subarachnoid space. Subarachnoid space filled with cerebrospinal fluid. So, brain has 3 parts: forebrain, midbrain and hindbrain. Forebrain also known as progencephalon. It is further divisible into 3 parts: cerebrum, thalamus and hypothalamus. Cerebrum contains 2 cerebral hemispheres: right cerebral hemisphere, left cerebral hemisphere. The 2 cerebral hemispheres are together known as cerebrum. Cerebrum is 2/3 of the total brain. Outer part of the cerebrum is known as cerebral cortex. It is grey in color due to presence of Nissel's granules. You already know brain contains 100 billion neurons. So in the neuron, cyton is directed towards the peripheral part of the brain. So numerous cytons, billions of cytons. In cerebral cortex, 10 billions. 1 billion is equal to 1000 millions. So 10 billion neurons present in the cerebral cortex. Nissel's granules present in the cytons of 10 billion neurons, giving grey appearance. Cerebral cortex. Okay, each cerebral hemisphere contains 4 lobes: anterior frontal, parietal, temporal and posterior occipital. So each cerebral hemisphere has 4 lobes: frontal lobe, parietal lobe, temporal lobe and occipital lobe. The surface of cerebral cortex has several folds. These folds are known as gyri. Deep cavities known as fissures, shallow cavities known as sulci. Gyri and sulci responsible for increase of surface area of cerebral hemispheres. Cerebral cortex contains 3 parts: sensory area, motor area and associated area. Associated area performs special functions like memory and communications. So next point is outer part of the brain, the particularly cerebral hemispheres. Outer part of the cerebral hemisphere is known as cerebral cortex, grey in color. Inner surface of the cerebral hemispheres, white in color, known as medulla. So outer cortex, inner medulla. Reverse in spinal cord. In spinal cord, outer white matter, inner grey matter. The medulla of the 2 cerebral hemispheres connected by corpus callosum. Corpus callosum. Inner surface of cerebral hemispheres contain lobes: amygdala and hippocampal lobe. Amygdala and hippocampal lobes together known as the limbic system. Limbic system control the sexual behavior and emotional behavior. Cavity is present in the brain known as ventricle. The ventricle present in right cerebral hemisphere known as first ventricle. The ventricle present in the left cerebral hemisphere known as second ventricle. Ventricles of cerebral hemispheres also known as paracells or lateral ventricles. The paracells present in the cerebral hemispheres open into diocele present in the thalamus. The two cerebral hemispheres separated by longitudinal fissures. Longitudinal fissure. Next one, thalamus. Roof of the thalamus is known as epithalamus. With the help of pineal stalk, pineal body attaches to roof of the epithalamus. Base of the thalamus is known as hypothalamus. From the hypothalamus, a stalk arises known as infundibulum. With the help of infundibulum, pituitary gland attaches to hypothalamus. So hypothalamus considered as the master endocrine center. Pituitary gland considered as master endocrine gland. Cavity present in the thalamus is known as third ventricle or diocele. Hypothalamus controls the autonomous nervous system, center for temperature, pain, thrust and satiety. Satiety means satisfaction. So these are the three parts of forebrain: cerebrum, thalamus and hypothalamus. Next one, midbrain. Midbrain also known as mesencephalon. Midbrain is in the form of four optic lobes on the dorsal surface. The anterior two optic lobes are bigger, known as superior colliculi. Posterior small are known as inferior colliculi. Superior colliculi responsible for vision, inferior colliculi responsible for hearing. On the ventral surface, two longitudinal tracts arise from the midbrain. They travel anteriorly and posteriorly. These longitudinal tracts are known as crura cerebri. They travel anteriorly connected to forebrain, travel posteriorly connected to hindbrain. In this manner, crura cerebri of midbrain connect forebrain and hindbrain to midbrain. Next one is hindbrain. Hindbrain also known as rhombencephalon, divisible into three parts: cerebellum, pons varolii, medulla oblongata. Cerebellum is the second largest part of the brain, so it is also known as small brain. Cerebellum maintains the balance. Cerebellum has three lobes. Central lobe is known as vermis. On either side of vermis are two lateral lobes known as cerebellar hemispheres. Each cerebellar hemisphere again has three lobes: anterior lobe, posterior lobe and lateral lobe. Lateral lobe also known as floccular lobe. The white matter of cerebellum is in the form of a tree known as arbor vitae. Cerebellum acts as a relay station. So pons varolii act as a relay station. So what is the function of cerebellum? Cerebellum maintains the equilibrium. Next one, pons varolii. Pons varolii act as a relay station between cerebellum and the remaining part of the brain. Similarly, as a relay station between cerebellum and spinal cord. Pons varolii contain pneumotoxic center. Whenever the concentration of hydrogen and carbon dioxide concentration increases, at the time pons varolii increase the breathing rate, eliminate the excess amount of carbon dioxide and hydrogen cations. Next one is medulla oblongata. Medulla oblongata control the involuntary actions, swallowing. Similarly, control the involuntary actions of cardiac muscle, involuntary muscles, lungs, kidney, heart. So this is about brain, structure of brain. Next question is, compare the following: central nervous system and peripheral nervous system. Central nervous system control the remaining parts of the nervous system. Peripheral nervous system, cranial nerve or central nervous system represented by brain and spinal cord. Peripheral nervous system, cranial nerves and spinal cord. Brain and spinal cord together known as central nervous system. Cranial nerves and spinal nerves together known as peripheral nervous system. So central nervous system is the control and commanding center. So peripheral nervous system carry the information from brain to target organ and send messages from target organ to brain. Send messages from brain to target organ. Send impulses or information from various organ to brain. Resting potential and action potential. Resting potential and action potential. Polarized axon means the electrical potential before it receives the stimulus known as polarized unexcited axon. Inner surface is electrically negative, outer surface is electrically positive. So what is resting membrane potential? Electrical potential of an axon at rest is known as resting membrane potential. Resting membrane potential is inner surface of axolemma minus 70 millivolts. Action potential. Once the axon receives the information, once again, action membrane potential. Whenever the axon is stimulated, in that part for few milliseconds, electrical potential changes. It is known as action potential. Momentary change of electrical potential is known as action potential. Electrical potential of axon at rest is known as resting membrane potential. Whenever axon receive, in that part, momentary change of electrical potential. So momentary change of electrical potential, so due to stimulus, is known as action potential. Next one is, so choroid and retina, so they are related to eye, not included. Explain the following process. Polarization of the membrane of a nerve ring. Polarization, so simple. Polarization, polarized axon or polarized nerve means unexcited neuron, unexcited axon. Electrical potential of unexcited axon, that is polarized. In polarized axon, inner surface is minus 70 millivolts. Just outside the axolemma, the electrical potential is plus 70 millivolts. It is maintained by sodium potassium pump. Sodium potassium pump present in the axolemma. It sends 2 potassium into the exoplasm, 3 sodium into extracellular fluid. Sodium potassium pump maintain the electrical potential at rest. Due to presence of sodium potassium pump in a polarized axon, inner surface is minus 70 millivolts. Depolarized. Same point, whenever the axon receive that particular part only for few milliseconds, inner surface is electrically positive and outer surface is electrically negative. Observe, the remaining part is inner surface electrically negative, outer surface electrically positive. It is known as depolarization. Whenever the polarized axon receives stimulus for few milliseconds, electrical potential changes. Inner surface is electrically positive, outer surface is electrically negative. The information is present in the depolarized part. From the depolarized part, sodium ions move to the succeeding part. Along with the sodium ions, information also pass to succeeding part. Again, the polarity reversed. So again, what happened after depolarization, in the same part, inner surface is electrically negative. That is known as repolarization. So here, simple definition, depolarization. Whenever a polarized axon receives the stimulus for few milliseconds, inner surface is electrically positive and outer surface is electrically negative. It is known as depolarization. Next one, conduction of a nerve impulse along a nerve fiber. Conduction of nerve impulse along the nerve fiber. Whenever the polarized axon receives the stimulus, you already know sodium voltage gate channels present in the axolemma. Sodium voltage gate channel, gates of sodium voltage gate channel open. Abundant amount of sodium enter along with the positive charge. Information also enter. So due to entry of positive charge, negative charge decreases. Minus 70 become minus 50, minus 20, 0, plus 20, plus 45. In this manner, inner surface is electrically positive and outer surface is electrically negative. That part is A. In the succeeding B, in the succeeding part, it is not electrically excited. That's why inner surface is electrically negative, outer surface is electrically positive. Now current flows from A to B inside the axon and current flows from B to A outside the axon. So a complete circuit is completed. So due to movement of A to B inside, B is electrically inside, B is electrically positive. In the A, outside is electrically positive and inside is electrically negative. So in this manner, so depolarization is conducted from A to B inside the axon. So later it is travel from one end to another end, conduction of nerve impulse from one end to another end of the same axon. So next step, from B to C, same procedure. Inner surface, positive charge move from B to C. Outer surface, positive charge move from C to B. In this manner, action potential is conducted from one end to another end. So next question is transmission of nerve impulse across a chemical synapse. Across a chemical synapse. Information or message travel from one neuron to another neuron known as synaptic transmission. Empty place present between axon terminal of one neuron, dendrites of another neuron is known as synapse. The membrane cover the axon terminals known as presynaptic membrane. The plasma membrane of dendrite and cyton known as postsynaptic membrane. Presynaptic membrane, synapse, postsynaptic membrane, together known as synaptic cleft. Together known as synaptic cleft. So synaptic transmission. Transmission of information or message from one neuron to another neuron known as synaptic transmission. Empty place present between axon terminals of one neuron, dendrites or cytons of another neuron known as synapse. The terminal part of axon terminal are swollen known as synaptic knobs. Synaptic knobs contain synaptic vesicles. Synaptic vesicles store the neurotransmitters. Synaptic vesicles store the neurotransmitter. Synapses are of two types, chemical synapses and electrical synapses. Electrical synapses are rare in animals. Most of the synapses are chemical synapses. Here chemical means neurotransmitter. Neurotransmitter is required for the transmission of information or message from one neuron to another neuron. Neurotransmitters present in the synaptic vesicles only. So in dendrites no neurotransmitter. So information always in chemical synaptic transmission, information always transmitted from axon terminals of one neuron to dendrites of another neuron, unidirectional. In case of electrical synapses, synapse is a small gap or sometimes no synapse between axon terminals of one neuron and dendrites of another neuron. No need of neurotransmitter. So presynaptic neuron transfer the information to postsynaptic neuron. Postsynaptic neuron also transfer the information to presynaptic neuron. Bidirectional transmission of information occur in electrical synapses, but electrical synapses are very rare in animals. Now we discuss synaptic transmission, means chemical synaptic transmission. When the axon terminals are electrically positive, at the time calcium voltage gate channels present on the presynaptic membrane open allow the calcium ion. When the calcium ion concentration increases, synaptic vesicles move to periphery, burst and release the neurotransmitters. Along with the neurotransmitters, information or message reach the ligand gated channel, ionic channel present on the dendrite and cyton, known as ligand gated channels. The ligand gated channels open. The neurotransmitters are of two types: excitatory neurotransmitter and inhibitory neurotransmitter. Common neurotransmitter is acetylcholine, means abundant. Others are norepinephrine, dopamine, serotonin. They are act as either excitatory or inhibitory neurotransmitter. If a neurotransmitter act as excitatory, the ligand gated allow sodium ions. If it acts as a inhibitory, the ligand gated channel allow the chloride ions. Two neurotransmitters always act as inhibitory neurotransmitters: gamma aminobutyric acid and glycine. They always act as inhibitory neurotransmitters. Summation of ions, addition of ions occur at the axon hillock. What is axon hillock? The part of the cyton from which axon arise, known as axon hillock. For example, 1000 sodium ions, 750 chloride ions. So positive charge is more. At the time postsynaptic neuron is depolarized and carry the information to successive neuron. So sodium ions are known as excitatory postsynaptic potentials. Chloride ions are known as inhibitory postsynaptic potentials. So the depolarization or repolarization depends upon excitatory postsynaptic potentials and inhibitory postsynaptic potential. If excitatory postsynaptic potentials exceed inhibitory, the postsynaptic neuron is depolarized and carry the information. If the inhibitory postsynaptic potential means chloride ions are more than sodium ions, the postsynaptic neuron is hyperpolarized, more negative charge, and it failed to transfer the information to succeeding or successive neurons. So synaptic transmission. Next one is draw label diagram of the neuron and brain. Eye and ear are not included. Write short note on the following: neural coordination, forebrain, midbrain, hindbrain. We already discussed. Neural coordination. The neurons control and coordinate any organ within few milliseconds, but action is short-lived. Reason is the neurons never transfer the information or message to each and every cell, transfer the information to organ or tissue, but endocrine system transfer the information along with the hormones to each and every cell, between the cells, intercellular spaces. That's why effect of endocrine system is more long lasting when compared to neural system. Mechanism of synaptic transmission, we already discussed. So mechanism of vision and hearing not included. Seventh, how do you perceive? So that is which part? Seventh B is included in your syllabus. Which part of our body help us in maintain the body balance? So body balance by cerebellum and internal ear. How does the, I not included. Explain the following. Role of sodium in the generation of action potential. So along with the sodium ions only information enter. So through sodium voltage gate channel, sodium enters. As a result, inner surface is depolarized. Mechanism of, differentiate between myelinated and non-myelinated neuron. Myelinated neuron, Schwann cell or oligodendrocyte secrete myelin sheath. In a myelinated neuron, information jumps from one node to another node. So myelinated neurons exhibit saltatory conduction. Saltatory means leaping. Non-myelinated neuron, continuous conduction. Each and every part conduct the information. Next point, myelinated neuron transfer the information 50 to 100 times faster than non-myelinated neurons. Dendrites and axon. Dendrite and axon are the two associated process of neuron. Dendrite is small. It is anterior to cyton. The function of dendrite is receive information from the neighboring neurons or surrounding environment and transfer the information to cyton. That's why dendrite is known as afferent process of the neuron. Axon is long. It is present behind the cyton. Axon always carry information away from the cyton. That's why axon is known as afferent process of the neuron. Thalamus and hypothalamus. Thalamus is the posterior part of the forebrain. It control and coordinate the sensory and motor signaling. Base of the thalamus is hypothalamus. Hypothalamus regulate the body temperature, hunger, thirst, pain and satiety center, satisfaction center. Cerebrum and cerebellum. Cerebrum is the largest part of the brain. Cerebellum is the second largest part of the brain. Cerebellum also known as small brain. Cerebrum regulate the voluntary actions, intelligence, memory, thinking, attitude, and cerebellum maintain the balance. Which part of the human brain is the most developed? 10th, A is not included. B, which part of the human brain is the most developed? That is the forebrain. Which part of our central nervous system act as a master clock? So master clock, pineal body, particularly a optic chiasma, suprachiasmatic nucleus is present on the optic chiasma, act as a biological clock. Pituitary gland control the biological clock. So in general usage we may say, pineal body, not pituitary. Pineal body is the biological clock. It produces melatonin hormone. So biological clock is pineal body. It is present on the dorsal surface of the thalamus. You all know with the help of pineal stalk, pineal body attaches to epithalamus. Next one is fovea iris, so all related to eye, ear, not included. Afferent neuron and afferent neuron. 12. Afferent neuron carry information from receptor to brain. Afferent neuron carry messages from brain to target organ. So impulses conduction in a myelinated nerve fiber is saltatory movement, jumping. Information jumps from one node to another node, known as a saltatory movement. And myelinated nerve fiber carry the information 50 to 100 times faster than non-myelinated nerve fiber. Cranial nerves and spinal nerves. Nerves associated with the brain, cranial nerves. Nerves associated with the spinal cord, spinal nerves. So mammals have 12 pairs of cranial nerves, 30. So human being 31 pairs of spinal nerve. So cranial, all spinal nerves are mixed, but cranial nerves, some are sensory, some are motor, some are mixed. You already know, first, second, eighth are sensory, third, fourth, sixth, 11th, 12th are motor, fifth, seventh, ninth, 10th are mixed cranial nerves. So next class we discuss chemical coordination and control and chemical control and coordination.